Hello, my name is Melissa Griffith, and today I will be presenting on DBCP and male infertility in Lathrop, California, United States. The main purpose of this presentation is to describe the DBCP contamination in Lathrop, evaluate the effects that it had on the people involved, and assess the long-term impacts of the incident. Throughout this presentation, I will first discuss the main use of DBCP and why it was manufactured. Following this, I will show the impact of DBCP on workers at the Occidental Chemical Plant in Lathrop, specifically the effect that it had on their fertility. I will then present the data that was available before the incident about its effects on rat sperm production, as well as carcinogenic effects on rats and mice. I will finish by talking about the long-term impacts of the incident, what was learned, and what is still harming us today. DBCP, or dibromochloropropene, was found to be effective at exterminating nematodes, a parasitic worm that infects the roots of citrus plants. A nematode's life cycle is entwined within the roots of these plants. They begin by hatching from eggs, slaving the soil, where they quickly find a root to begin draining nutrients from. Once they grow enough, the mature female finds a mature male to mate and produce more eggs. Spreading the infection, this can cause significant damage to the roots of citrus plants. DBCP was introduced into U.S. agriculture in 1955, and it was approved for use as a fumigant in 1964. The compound was widely marketed and became a commercial success. In 1977, an emergency study by the EPA agency discovered that workers at Lathrop were suffering from deficient or absent sperm. Controls were improved at U.S. facilities, but the product continued to be marketed and sprayed in Latin America, the Philippines, and some African countries. Unfortunately, there were serious downsides to the use of DBCP, as demonstrated by its effects on the workers at the Occidental Chemical Plant at Lathrop, California, a manufacturing plant for DBCP. In 1977, many of the employees at the Occidental Chemical Plant who had handled DBCP were found to be either azoospermic or oligospermic. Subsequent surveys of employees handling DBCP at other chemical plants confirmed these findings. As you can see, the difference between exposed and non-exposed workers is staggering. The amount of exposed workers that were totally sterile or azoospermic was 13.1%, compared with only 2.9% of those not exposed. Similar results were found with those with extreme depleted sperm counts. The differences between unexposed and exposed workers is significant, representing a drop from over 90% of workers with normal sperm counts just over 50%. The results from these tests were so severe that OSHA pushed to create a new emergency temporary standard of 1 parts per billion. Next, we are going to take a look at the effects of long-term exposure of DBCP on rat sperm production based on the results from a 1975 study. What makes this incident particularly interesting is the fact that there was already data available about DBCP's effect on fertility as early as 1961. Taken from a study in 1975, we can see the effects that DBCP has on rats' sperm levels after long-term exposure. By 2 months into the experiment, there was already a disparity of slightly less than 1 million sperm cells per milliliter, which then increased to over 2 million after another 2 months. This decrease in sperm count was followed with the shrinking of the testes, eventually leading to necrosis. This clearly demonstrates the potential for DBCP to cause infertility. In 1972, the National Cancer Institute initiated a study to ascertain the possible carcinogenic activity of DBCP as well as other pesticides. Via oral intubation, male and female Osborne-Mendel rats and hybrid B6C3F1 mice were fed DBCP at 2 dose levels, 15 and 30 mg/kg per day for the rats and 130 and 260 mg/kg per day for the mice, for 5 days per week, for a total of 78 weeks. As early as 10 weeks after the studies began, squamous cell carcinomas of the stomach were detected in both male and female rats and mice. At the termination of the studies, stomach cancer was noted in more than 212, or 60% of the rats, and 90% of the mice. In addition, the stomach tumors readily metastasized to a wide variety of organs, particularly to the lungs, and a high incidence of mammary gland endocarcinoma was noted in female rats, but not in female mice. The NCI bioassay program labeled DBCP as very carcinogenic. Several of the investigators involved in the research have published these studies. There were several significant impacts of the DBCP incident in Lathrop. First, the widespread attention of this incident received led to many other places producing DBCP to test its workers, finding similar and sometimes even worse results. This allowed for these workers to be isolated from the contaminated workspace and added to the string of class action lawsuits which encouraged further regulations on the production of the chemical. Provided new information which informed the emergency temporary standard 1 part per billion that OSHA put into place eventually became a permanent standard. Further, it led to the EPA analyzing the impacts that DBCP had on farm workers where the chemical was fumigated into the fields. Eventually, DBCP was banned altogether in the U.S., as we discussed earlier, except for Hawaii. It's worth noting that it was not banned in other countries, where we discussed it had been shipped out to parts of Africa and South America, later leading to infertility issues from the outsourcing of the pesticides to countries less stringent on DBCP regulation. It is also important to note that the production and use of DBCP as a pesticide had long-lasting impacts in the water of the areas that were exposed. With levels above the acceptable limit in over 18 states due to the chemical's low decomposition rate, this shows the long-lasting impact that improperly regulated pesticides can have due to its ability to leak into the water table. There are many lessons that can be learned from the incident in Lathrop, California. We know that DBCP was determined to be a human reproductive toxicant causing azoospermia and oligospermia, thus making it inappropriate for use as a pesticide. Long-term exposure can increase the risk of testicular toxicity. Therefore, many class action lawsuits were filed against American chemical companies. The impacts of DBCP are long-lasting, and therefore increased regulation of existing and new chemicals is necessary to avoid catastrophes such as the one in Lathrop, California.